the new microphone. It's so exciting. Welcome to the Arnold Pfeffer Center for Neuropsychoanalysis at the New York Psychoanalytic Society and Institute. This is our last meeting of this academic year. Does it sound normal? It sounds sort of tinny to me. Does it sound good? It sounds handsome. So it's our last meeting of the year. Thanks for giving up a little bit of your summer to come and learn about the dynamic memory trace from Cristina Alberini, who's very clever. She has degrees from Columbia and Pavia and Genoa in neuroscience, biology, immunology, everything. She's professor of psychiatry at Mount Sinai and professor of neuroscience and professor of chemistry and biology. And she's lovely and clever, and we're now going to learn about the dynamics of the memory trace. She's doing very, very, very cutting-edge work on this whole reconsolidation thing, and I, for one, can't wait to hear what she's got to teach us. And she's an analyst, training in analysis, believe it or not.
Thank you very much. Now you got such high expectations that I don't know how I'm going to do that. It's a great opportunity to be here. Thank you for the invitation. Can you hear me in the back? Is it good? Okay. It's a great opportunity to discuss what is going on in the lab, what is going on in the field. There are many exciting questions about how memories are formed, stored, and retrieved. And that's important not only in neuroscience and medicine, but also in psychology, psychoanalysis, learning, education. So it's not just a neuroscientific question that we are asking. We're asking at the neuroscience level, but I hope today we're going to have an opportunity to discuss something that can be important also for psychoanalysis. I'm going to start with a couple of introductory slides so that we're all on the same page. Memories come in different shapes and forms and types, and scientists like to classify. So a couple of classification divides memories depending on the duration first, and that definition classifies memories as short-term or long-term memories. Short-term memories are the memories that last for minutes, and long-term memories are the memories that last for days, weeks, months, even a lifetime. And those two types of memories are very different in terms of mechanism. We're going to discuss today about long-term memories. This is what we are interested in the lab. An example of short-term memory. You have to memorize a telephone number, so you have to make a reservation, you memorize the number, you make the call, and after the call, that memory is gone. We don't need to keep that information, so our brain doesn't keep it. But sometimes things happen that are very important. They have to be important. And they are important because biologically speaking, they are important. We're going to see some examples. And so the brain has evolved a number of mechanisms so that those information are going to be maintained as long-term memories. Then among long-term memories, we have different types of memories. And we all experience long-term memories that last for days, months, sometimes even a lifetime, even if they have been encoded or formed just after a single episode. Those are the longest memories. And we're going to see why those are very long lasting. So then, as I said, among the long-term memories, we have different forms. And one distinction divides these long-term memories in two big groups: explicit or declarative, and or implicit or non-declarative. And the distinction is based on the so-called memory system that they use, meaning the circuitry, the brain regions that are involved in the formation and the storage of this type of memories. So the explicit or declarative memories are the memories of facts, people, things, the memory we can talk about, our history. And these memories use the medial temporal lobe. That is a region that is placed behind our ears. And we know that because when this region is damaged, this type of memories are impaired. So people cannot form new memories. They lose the memories that has been formed previously up to a certain point. So this region is medial temporal lobe, and particularly a region of the medial temporal lobe known as hippocampus, is very important for the formation of these long-term memories. And the storage for some time, for a limited time, is also important for the storage of these memories. And then we have implicit or non-declarative memories. These are memories of how to do things. There are memories that the recollection of it is not conscious anymore. After the learning has occurred, they become sort of automatic, subconscious. And this is how to do things, for example, how to ride a bike, drive a car, tie your shoes, this type of memory. And they include a number of different types, but they all have this quality. It's not about facts, people, and things, it's not our history. So the explicit or declarative memories are the memories that we are studying in the lab. We study model of it. We don't study human memories. We study this type of memory modeled in an animal task. So we study hippocampal dependent memories. That's how we reproduce this type of memories in an animal model. And what's important also about these memories is that these are the memories that contribute to our identity. In a way, we are who we are because of our memories. We lose our memories, we lose ourselves. And that's why it's so devastating. Diseases like losing memories, Alzheimer's disease, neurodegeneration that lead to explicit memory loss is very frightening. And obviously these are some of the reasons why in the lab we're interested in understanding how does the brain make these long-term memories? How can we maintain the information? Can we find some way to prevent or cure this disruption of memories in Alzheimer's disease or neurodegeneration or even memory loss over age? I'm going to show you a couple of videos. This is an image that shows where the temporal lobe is. As I said, these memories require this region of the brain. If it is damaged, we cannot form new long-term memories, and we're going to lose some memories that have been encoded just before the damage. There is a gradient of loss. So we're going to see an anterograde amnesia, the loss of the new memories, and a retrograde amnesia, the loss of the memories that occurred before the damage. And I'm going to show you a couple of videos that really exemplify how difficult and devastating life can be without long-term memory. The first one describes a case known as EP, who suffered from an acute viral infection. And after the viral infection, the brain remained damaged, and particularly the medial temporal lobe. And you're going to see how difficult it is for him to encode new memory, to retain new information. And at one point he keeps repeating himself because he forgot that he just said what he said. The second case is Chuck, younger case. He suffered from a heart attack, and that led to damage of the hippocampus. And then we're going to see both how difficult it is for him to encode the memory, and also how much he lost the memories of previous events of his life. The interviews are by Alan Amada.
Today, Larry Squire and Jen Frascino are on the latest of many visits the team has made to the man known to memory researchers everywhere as EP.
Oh, wonderful. Got a nice son for the camera.
Yeah. EP is now 82. In 1992, retired from a career in electronics, he suffered an acute virus infection that destroyed part of his brain.
Research and development, that's what I like.
Most of his thinking skills survived the damage virtually intact.
Why should people pay taxes?
To support their country and their counties and states to pay for things.
If you were lost in the forest during the day, how would you go about finding your way out?
I would find a river and follow the river down to wherever it may go. Ocean, lake.
EP can also copy complex drawings. And he not only has no difficulty repeating back a list of words, in this case, dog, raccoon, lion, and parrot.
Dog, raccoon, lion, and a parrot.
Good.
But he even has a little sly fun with Jen along the way.
What you gotta add is about 2 more.
Oh, really? Would that make it tougher?
And that would catch them. 7, you know. Go. Having been in electronics over the years, I am amazed at all that. It's just wonderful. Believe me, little printed circuits, just, oh, you can't believe it. It used to be in a 6-foot rack, and down to this, it's just wonderful. When I was in electronics, this would be in a 6-foot rack.
But it's now that something odd about EP's behavior becomes apparent.
Printed circuits, diodes, triodes, and all that stuff in there. Oh, it's just amazing, believe me. When I was in electronics, this would be in a 6-foot rack. This would be in a 6-foot rack. It'd be in a couple of 6-foot racks. It'd be in 6-foot racks. This would be in a 6-foot rack. And now look at it, down to this. Little printed circuits. And
So he's not aware that he's told us 3 or 4 times that he used to be in electronics, that a computer used to fill a room, and that now it's down to the size of a microchip. He's just not aware that he said those things several times.
No, he tells us the same stories again and again. We've heard these stories before, and we'll hear these stories again.
I forget her name. What's her name?
her name?
Yeah.
I don't remember.
Okay, Jennifer.
Jennifer. Yeah.
I'm sorry.
Have you met Jennifer before today?
I think so, yes. I'm not positive though. I'm sorry,
No.
Oh, that's okay.
Okay.
We all have.
Well, we all just have.
Oh, sure. How much have you worked with him?
We have probably visited his house 200 times.
200 times. And for him, is each time the first time for him?
Well, you see, it's yes and no. It's clear that he does have a record of the experiences that are based on the habits, the interactions, the emotional values of the interactions, the fact that they've been positive and not negative.
The second video.
Then we're meeting another amnesic. But this time it's a man whose hippocampus, although badly damaged as a result of a heart attack 5 years ago, still struggles to put memories together.
So what I'd like to do is just show you some pictures and ask you, what can you tell me about these pictures? Do they evoke any memory? What does it bring back for you?
Chuck's memory is being studied by Dr. Mika Verfeli.
Here, this is something that you might have seen in any of the local newspapers in the last year.
It certainly has something to do with people objecting to.
We're in Boston, where the recent sex abuse scandals in the Catholic Church have received intense media coverage.
People who are somehow hurting, abusing, not paying enough attention to others.
Good. Is that a picture that looks familiar to you?
I believe this is a picture taken the day or days after the New York towers in New York were all...
Chuck, do you remember where you were when that happened?
Not at all. I remember at the time being especially worried because my son was in New York, and obviously the first thing...
As Chuck's wife remembers very well, this isn't so. Their son lives in New York today, but he didn't on 9/11. Chuck taught English before his illness, and he's continued to write since, giving us a unique glimpse into a mind that knows only too acutely what it has lost.
I cannot remember my wedding day or the celebration surrounding it. I do not recall the birth of my sons, my father's death, or who most other family members are. There are many.
When we first came in here this morning, we actually started off with looking at some pictures.
Right.
Do you remember?
Can I remember that?
Right.
Let me think, please. I'm just getting glimpses. Not even any shapes or actual depictions of anything that was there.
Chuck certainly has severe damage to the hippocampus, but his hippocampus isn't gone completely. So there's still something working to some extent. And what happens is that some experiences, some fragments of experiences may stick, but they're not linked in the correct way. They're not linked to other things that happened. They're not linked to the correct context. Sometimes they're not linked to the correct people. And so in that way, we're left with fragments that are not integrated. Occasionally, these fragments will pop up. They may pop up in seeing a picture, but as he described it, he may also have sort of random thoughts and not know, is this really a memory or is it just a thought I'm having?
So, we have seen how difficult it is life without memories, how important is long-term memory. Memories protect us from danger. That's one of the main biological important functions. So that we don't repeat mistakes, so that we don't get in danger anymore, so that we protect our species. But also memories are important to find the best sources of food, the best sources of sex, to have the most adaptive life possible. So obviously we want to know what makes a memory long-lasting. And we are looking at this type of memories, episodic, declarative, or temporal lobe dependent. One important mechanism that allow newly learned experience to become a long-term memory is the emotional charge. We have seen these examples. They show, as you know, to test long-term memory, these examples of traumatic memories, very emotionally charged memories, and those are the ones that we remember for a life. What happens? Where were you when you heard about September 11th? You obviously remember that very well, with a number of details, the people, the sound, the smell. Many information become long-term memories when the emotion is very strong. So we know that, from experience, we know that from a number of studies, in both humans and animal models, that emotionally charged or salient events are better remembered. And obviously there is all a gradient. The traumatic memories are going to be much longer lasting. The ones salient but not as traumatic are going to be less lasting or less detailed. There is also one important thing to say here, that if the memory is too traumatic, then there is actually the opposite effect. In fact, the strength of the emotion or of the stress of the trauma leads to an inverted U-curve, we call, in terms of the memory retention or performance. So some stress will lead to a long-lasting memory up to a certain point. If it is too much, then actually there is no memory. There is forgetting, or there is memory loss. It's not clear why, whether the memory is never formed, or whether our brain represses the remembering of those episodes. But certainly, in order to become long-term memory, the information that has been processed is linked to some sort of emotion. That we know. So it can be obviously positive or negative, and sometimes it can be extreme, like a traumatic experience. And I'm not going to talk about much in psychoanalytic terms, but I wanted to mention that the first one who really elaborated about memories, and especially memories of traumatic memories, was Sigmund Freud. And he wrote tons about how memories are processed in the brain, how traumatic memories are different than normal memories. And here I'm going to read just this sentence, which exemplified to me, during my training, I was amazed to see how much he was able to talk about memories, not knowing the mechanisms or not much about how the brain processes memories in his days, things that we are finding out now in neuroscience 100 years later. So this is the letter to Fries in 1896, where he says, "Our psychic mechanism has come into being by a process of stratification. The material present in the form of memory traces being subjected from time to time to a rearrangement in accordance to fresh circumstances, to a retranscription. Memory is present not once, but several times over." Back to neuroscience. What we know in terms of the process that leads to the newly learned information becoming a long-term memory, we know that it takes time. It's a complex process that is defined as memory consolidation. And the reason why they use these terms actually comes from studies more than 100 years ago in humans, verbal learning, in which they tested subjects and they gave them two lists of words to learn, one after the other. This was Müller and Pilziker around 1900. And the subject could learn very well the two lists of words if they were separated by a certain interval of time. But if they were given one after the other, they could learn better the second list. They could remember better the second list of words. But the first one, there was some sort of forgetting and interference. And they found out that time is what makes the information that is processed transform that into a long-term memory, which remains in a more correct way in its retention. So the brain needs time after encoding to transform this information into a long-lasting memory. They said to be fixated in the brain or consolidated. That's how the term consolidation came up. So the consolidation process is the process that takes this newly learned information, which is in a labile state because it can be disrupted by interferences, and transform it into a long-lasting memory that is resilient to disruption. And then over 100 years, many studies have been done on this consolidation process. And it was found that there are many different types of interferences that can disrupt the consolidation process. We have, for example, pharmacological interferences. A number of blockers of certain types of biological mechanisms will interfere with the consolidation if given during that time window. Many studies have been done particularly with protein and RNA synthesis inhibitors. Those are blockers of the making of new protein and messenger RNAs. So we know that long-term memories require those new proteins and new messenger RNAs. So it requires the expression of our genes in the brain. If we block that, long-term memories do not form. We know that if we inactivate the brain, we see the same thing, obviously. You have seen clinical examples where the brain is not active, some part of the brain, the hippocampus, is inactive because of pathological degeneration or blocking. And long-term memory consolidation is interfered. Even the temperature, by cooling the tissue, we can interfere. Seizures, those are clinical examples. People who have seizures, they show interference for long-term memory formation of the events that just occurred before. Brain trauma, and even new learning, as I said, some of the experiments have been done and shown that new learning interferes with the consolidation of whatever has been just presented. So that is actually a behavioral type of intervention that we could use to block the consolidation of certain type of memories. We can use new behavior, new learning, something that can interfere with the consolidation of what just happened. It's not just pharmacology. So the important thing is that we know that it takes time. It requires this new gene expression that we can use in the lab. And we want to frame what happens over time. What are the mechanisms that change over time in the brain that by the encoding of new learning transform this new learning to a long-term memory. So the classical consolidation hypothesis that came out from all these studies suggested that this consolidation process is only one for whatever we learn, meaning it's a unitary process. We learn something new, that experience remains in a labile state for some time, so we can interfere with its consolidation, but over time it becomes resilient to disruption. It becomes consolidated, and the process is a unitary process that occurs over time. It turns out that that's not the case. More recently, and particularly in the last 10 years, and this was really a reawakening of the field in the year 2000 by the studies done at NYU by Karin Nader, Len Schaaf, and Joe Ledoux, which really re-examined a number of results that have been actually described first in the 60s, but they were never followed up because people didn't believe it was true. There was some sort of disagreement in the field, and those data basically disappeared from the face of the earth. But around the year 2000, these experiments that I'm just going to describe at NYU in Joe Ledoux's lab, they revamped this important question because they show that it is not true that a memory that has become resilient to disruption, say for example by blocking new protein synthesis and therefore consolidated, it is not true that it's going to remain consolidated forever. That memory can become labile again if it is recalled. So they took animal models and they used fear conditioning, type of task that is widely used in the lab because the animal learns in a single experience that something is important, is fearful, and they are going to show that memory, that long-term memory. So it's very easy to induce these memories, very easy to test it. I'm going to show you an example. So they found that if they give, the animal learns these memory, they have this memory, and they give some time to consolidate. Then they recall this memory in the lab, and they inject right after the recall these disruptors, these protein synthesis inhibitors, and then they test them again. They saw that the memory was gone, showing that that memory after retrieval became labile again and sensitive to disruption. And because the same type of interferences interferes with this process of restabilization of the memory after recall, they call that memory reconsolidation because it's reminiscent of the consolidation of a new memory. The same type of interferences that disrupt the consolidation as well as the reconsolidation of the memory. So they propose this hypothesis that memories, when retrieved, they become labile again, fragile again. So we can use some interferences to get rid of those memories, but if we don't do anything, actually memories over time stabilizes again, reconsolidate again. And therefore they are maintained. Turns out that that's not really the whole story. So we became interested in studying consolidation. We were already studying consolidation. We became interested in studying reconsolidation. And obviously, I don't have to spend too much time to convince you that this is important. For example, it is important that we know how memories are formed and stored and how we can change the strength of the memory. What happens over time to our memories? If we know that, can we disrupt memories that are linked to pathogenic states? So, for example, can we disrupt memories or components of the memory selectively so that we can weaken pathogenic memories, for example, those that are linked to post-traumatic stress disorders? Those are very particular types of memory, by the way. They're not normal memories of trauma. Only a fraction of the people, as you know, develop PTSD. But those memories are very strong, very vivid, intrusive, they interfere with the life of the subject. Phobias, depression, trauma, a number of pathologies that are linked to the memories that come back, traumatic memories, disturbing memories that come back. So if we have a way that can be pharmacological, but it can be also behavioral, to interfere with the consolidation or reconsolidation of the memories, we have an opportunity to decrease the intensity, in particular maybe the emotional intensity, not so much the content, hopefully, but the emotion attached to it. And this is, you are very familiar with this type of work in your everyday work. So can we change, in a controlled fashion, some of the components like the affect that is contained in our memories, and this is what we use in psychotherapeutic setting. So reconsolidation and memory fragility after recall. We're going to ask a couple of questions today. Why does memory become labile after recall? Certainly not because we need to use that in psychoanalysis. The biology has designed that. And what is the contribution of time? Are recent memories undergoing reconsolidation as much as the older ones? That's an important question as we consider our history and we want to work with memories that obviously are of different age. So to study these questions, we use a task that is known as inhibitory avoidance. And we use rats as species. And in this task, we use a shuttle box that has two compartments. One is lit, the other is dark. There's a door separating the two. We place the rat into the lit compartment, and then we open the door that allows the rat to go into the dark side. And in the dark compartment, the door closes, they get a foot shock, an electric foot shock. That is the learning. The training is completed. Obviously, they remember that very well. Who wouldn't? We go into a room, we get something unpleasant, or sometimes even more than unpleasant, shocking. Depending on the shock intensity here, we can reproduce the level of stress of it. It could be a mild experience, but it could be a more stressful experience. And after that single event, they remember very well. They form a long-term memory. So then we test the animals at whenever time we want to. And during testing, what we do, we replace the animal back into the lit compartment, open the door, and they obviously remember that if they get into the dark side, they're going to get shocked. So they're not going to go there. They're going to develop an avoidance. And so we measure the seconds that they remain into the lit compartment, the avoidance index. And so you are going to see some of the graphs I'm going to show you, to talk about the data we found, that are of these types. The acquisition bar, you see, is only a few seconds. Generally, it's around, the average is around 15 seconds. It's the time that the animal takes to enter into the dark side before they got the shock, during the acquisition phase. And after they got the shock, then they show a retention, a latency, they develop a latency of avoidance that is of many, many minutes. So our cutoff time is 540 seconds, 9 minutes. We cannot stay there all day because they don't go 5 minutes we take them back to their home cage. So that is the measure of our memory. And it's a pretty strong and robust memory index. This task, we know, requires an intact hippocampus and an intact amygdala, as well as an intact nucleus accumbens and other cortical areas. But the hippocampus and the amygdala are particularly studied because we know that the hippocampus is very important for the memories I told you about, facts, people, places, things. And the amygdala is important for the emotional memories. I'm sure you have heard several lectures talking about how amygdala is important for fear, emotion, stress. So we target in our studies to understand what type of biological changes occur in these regions. First, the hippocampus and the amygdala. To show you how reconsolidation works in this task, I'm going to show you an example of an experiment. The animals are trained, and 2 days later, I showed you that they have a nice retention, a nice memory, strong memory. 2 days later, they are tested. So the testing, for example, will give you an average retention of this level. This one here. Test one. Right after this testing, we know they have memory. The animals are divided in 2 groups, and one is treated with a protein synthesis inhibitors. That drug that blocks protein synthesis, which is required for reconsolidation. The other group is treated with saline, the control group. And then we test them 2 days later to see the effect. And what we found is that the group that has received an isomycin, the protein synthesis blocker, had a latency that was very low. The memory was almost gone. Whereas the group that received the saline injection, in the yellow here, they remain constant, as they should. Then we look at whether this effect is contingent upon the recall of the memory. So we give the treatment, an isomycin, here, without the recall of the memory at the same time point, and test again 2 days later. And what we found is that, in fact, there is a significant memory. It's somewhat lower than the controls, as you can see, and there is a reason for it, but it's certainly significantly higher than when we give the recall together with the drug. So these protein synthesis inhibitors, this drug, blocks the restabilization of the memory, and the restabilization has been induced by the retrieval, the recall of the memory. It's a classical reconsolidation effect. So we're going to ask, why do this memory become labile? And we have tested 2 hypotheses that have been proposed before, starting from the 60s, about the studies of reconsolidation. And the first hypothesis says, well, memory becomes labile, fragile, because we change the memories all the time. So when we retrieve our memories, because we need to retrieve our memories, and we encounter something new, and therefore we make a connection between the 2, otherwise our memories will be compartmentalized and we cannot make a continuous of what happened before and now and then. And so that's why it becomes labile, the old memory becomes labile, because it needs to incorporate new information. Therefore, it's used for memory updating. That's why memories become labile. And a second hypothesis says, well, memory actually reconsolidates because that's a way to strengthen memory. So we tested both hypotheses. And we also tested the contribution of time, the age of the memory. So we start with the first hypothesis. Reconsolidation mediates memory updating. And the way we reproduced that in the lab, it was to use the task I just described, adapted with a new circumstance, the recall of an old memory now presented with something new. And we had already identified in the lab some mechanisms that specifically are required for either the consolidation of a new memory or the reconsolidation of a retrieved one. So bear with me. Consolidation of a new memory, we know, requires the expression of a certain protein in the hippocampus. It's called CEBP beta, just for those of you who want to know. So if we disrupt this protein in the hippocampus, we disrupt consolidation of new memories. But nothing happens to the reconsolidation of memories that are retrieved. So it's a consolidation mechanism. And then we identify that the same mechanisms, but in amygdala, is actually required for reconsolidation and not consolidation. Meaning now if we disrupt this protein in the amygdala, we are going to disrupt reconsolidation of a retrieved memory. But the encoding of a new memory is not touched. So this allows us to selectively disrupt one or the other and ask the question of what is reconsolidation doing? What is consolidation doing? So we have designed the task using inhibitory avoidance and recreate a condition in which after the first learning, the animal is going to be presented with something new after the retrieval. And then we're going to ask the question, this new memory that is made of the old and the new, does it use reconsolidation? Is that why reconsolidation happens in the brain? So I'm going to go over again, which is the same thing that I just presented. There are two steps now. The rat is placed into this compartment, and then the light is turned on after a few seconds. And then a few seconds later, the door opens. The animal is going to go into the other room. The door closes, they get a foot shot. And they're going to learn to avoid that context. So that place we call context A, place A. Then some days later, we're going to place them in another room. Different walls, different floors, different smells, different room altogether. And what we're going to do, we're going to turn on the same light. The door is closed. They don't even know that there is something on the other side. Then the door is going to open. And if the light with the door opening is going to remind them of the old memory, of the shock, they're going to develop an avoidance for the new context when we open the door and test them. So we're going to test them in both contexts. So it's like as if we are in a place, there is this nice candle lighting on, then the door opens, we go into the other room, we see something shocking. Our parents having sex. We're going to avoid that. So then we go to another room, it's a completely different room, but there is the same candle, very strange candle that reminds us, oh my God, there is another room, maybe the same thing is going on, I'm not going to go in. But we never really experience that in the second room. We're just going to make a second association. We're going to update our memory. With the recall of an old one, the experience of a new place, and then we're going to make a new behavior. So, first of all, is it true that they develop an avoidance for the second room? In the second room. And that's the case. So there is a little bit of data here that I'm going to take you through because it's an interesting study, I think. So as I said, they're going to be trained in the first context, context A, with the light and the shock. Then 2 days later, they're going to be placed into this new room, context B. This is the reactivation of the trace of the memory. And they're going to be presented with the light. That's the cue that reminds them of the terrible experience they had in context A. And then we're going to test whether these animals are going to in fact develop an avoidance for context B. And the answer is yes. The red bar, those are the animals tested 2 days later, after they A have been trained in context A, reminded in context B with the like. The other two columns, groups that they have a very low memory, those are controls. Are the animals that have been tested in context A and remain in the home cage, so tested again in the context B. Actually, they are going to have no avoidance, they shouldn't. And the ones that have been exposed to the context B, but there was no light, there was no reminder of the trauma. And those, in fact, don't have any significant memory. Obviously, all the rats, if tested in the original context, they're going to show fear, because they have been trained, they have been shocked in the first context. So the blue bars are all high. Then we test them again a week later to see if this is a strong memory. Is it maintained over time? And we see the same thing. So yes, they do develop an avoidance in the second context if they are reminded of the shocking experience. And this is called second-order conditioning. Now we have the model to test our question. So the prediction is, if reconsolidation occurs to update the memory, to make this new connection between the reminding of the old shock with the new context, if that's the mechanism, by disrupting reconsolidation, we should get rid of the new memory. And we have a way to do that. As I show you, we know which mechanism we can use by targeting the amygdala. And we did that. So we disrupted that type of mechanism by injecting certain things in the amygdala of the animals that went through this procedure, and look at whether the new memory was disrupted. And what we found is that the memory for the first context A, was indeed disrupted by this interference, as it should be. We are disrupting reconsolidation. So the animals have been trained, they have been recalling the memory in another context, and then they get this interference. And then they are tested again. We know that this interferes with reconsolidation, and in fact, you see here, I'm going to move. The memory is lost because we have disrupted reconsolidation. The other column, this one, the third, is the one that received the same interference, but they didn't have the reactivation. So the recall, the retrieval of the memory has to be there in order for us to disrupt reconsolidation. But what was interesting is that actually the new memory, this one, was intact. This one here. This is the control, this is the interfere. So they lost the memory of the original context through reconsolidation, because that was disrupted. But reconsolidation did not interfere with the formation of the new memory, the memory for the second group. And this is the control that is not reactivated. So, what's going on? Is this a new memory? Perhaps this is a memory that consolidates. It's not a reconsolidation process. Actually, the two memories exist in parallel. There is an old memory that reconsolidates, but when it's retrieved and makes some new association, it forms another memory, a new one that consolidates. We can test that. So we interrupted the consolidation process in the hippocampus. We did the same experiment, but now targeting the hippocampus. And what in fact we found is that the reconsolidation of the memory, the blue, was intact. We know we don't interfere with that. But the new memory was lost. So the control is the high bar, and the one that we interfere is down here, towards 0. So this is a new memory. When we retrieve a memory and we reconsolidate that memory, we also have an opportunity to make new memories. But these new memories they are not induced, they are not created because of the reconsolidation. They are created because they are new memories. They consolidate as new traces. And this, I want to remind here of Freud. There are many traces. The memory comes back, not once, many times. The old memory reconsolidates, but also allows an opportunity to create new traces, new memories that coexist parallel. So you have probably heard, because reconsolidation has become very trendy, even in psychology and psychoanalysis, that reconsolidation mediates the updating, the rearrangements. Well, in our case, we found that that's not true. And if we think about that, also is logic, because if reconsolidation would do that, then we would change our memories continuously, and we will lose the old ones. So memory A is going to become memory B, but memory A is going to be lost. That's not what happens. What happens is that we have old memories, and then we can build on that with new ones. And now we have choices. That's what really happens. So reconsolidation is not used for that. Reconsolidation is used for something else. Consolidation is used to make new traces. And that's what happens in clinical setting. So the formation of new association, this type of updating, is not mediated by reconsolidation, but by new traces, the consolidation of new traces. The disruption of the new associations does not affect the stability of the old memories. So we can work through by making new memories. The old memories are there. So what happens? What does reconsolidation do then? We didn't answer the question. We know what it doesn't do, but we don't know what it does. So we're going to answer this question in a minute, but I have to introduce the concept of time. An important variable here is time. Are new memories behaving like old ones when they are retrieved? Are new memories or old memories reconsolidating in the same way, like the hypothesis that has been proposed suggests? So we can test that very easily. We're going to do our inhibitory avoidance task and retrieve the memory at different time points after training. Different age of the memory. The memory is going to be 2 days old, as I showed you. This is the experiment I already showed you. The animals are trained. 2 days later, the memory is recalled, retrieved, and then they get this interfering manipulation with an isomycin, the protein synthesis inhibitors, or obviously our saline controls. And what I showed you is that an isomycin disrupts the reconsolidation. So when we test again the memory 2 days later, there is virtually no memory, very, very little memory. That's contingent upon reactivation. If we don't give the recall, there is no significant effect. Now, when we do that to a 7 days old memory, we still see a significant memory disruption. Is this one here. Again, contingent upon reactivation. Saline controls are similar. But when the memory is 2 weeks old, there is no effect. When the memory is 4 weeks old, there is no effect. So time is very important if we want to use reconsolidation to change the memory strength, to disrupt the memories, and even to enhance the memories. I'm going to show you one example. So, does reconsolidation mediate memory strengthening? We're going to test the second hypothesis. And to do that, we're going to amplify a little bit the effect by giving more than one retrieval, more than one recall, so that we can see a bigger effect. So let's say we use 3 recalls. So we train the animals, and then we divide them into groups. One stays at home, do nothing. The other one is undergoes retrieval every 2 days, once every 2 days. And it's going to be a very short retrieval, very short recall of the memory. We're going to expose them to the lead compartment only for 10 seconds. The reason is that when the animal is exposed to the context without the shock, it's going to also learn something new. Not only is going to remember that the old memory happened there, it's also going to say, oh, this time didn't happen, I didn't get shocked. And if we repeatedly do that, and particularly with long exposures, we cause the so-called extinction. You're familiar with that. It's used in clinical treatments, for phobias particularly, and this type of thing. And it's very context dependent. So we want to avoid extinction, because otherwise we're going to see a decrease of the fear. But it's not what we want. Here we just want to remind them. So the shorter is the exposure, the less they extinguish, the more they reconsolidate. So that's why we give only 10-second exposure. They don't go into the dark side. They don't get any shock, obviously, and that's it. So after we give these 3 reminders, if we test them compared to the controls that remain in the home page here, or compare even to another group that was exposed for 10 seconds to a new context for the same amount of time every 2 days, there is a very significant memory strengthening. They avoid the place much more. So there is a much stronger memory. Now, is this due to reconsolidation? If it is, we should be able to disrupt this strengthening by giving protein synthesis inhibitors, the manipulation we know that disrupts reconsolidation. And so what we did was the following experiment. We're going to also test the contribution of time, not only the protein synthesis inhibitors. We're going to repeat the same experiment, but we're going to inject them with protein synthesis inhibitors or saline, and we're also going to test the same 3 reactivations given 2 weeks after training, when we know the memory is not labile, is not reconsolidating anymore. So we're going to do that. So this is what I just showed you. The 3 reactivations strengthens the memory compared to controls that did not receive them, and these are injected with saline. When we give the 3 retrievers 2 weeks after training, there is no effect. First of all, to begin with, the memory is a little stronger in the controls. So time does something to memory strengthening. And it became resilient to disruption. We know that, I showed you already. So there is no reconsolidation after 2 weeks. Now the question is, after the 3 reminder, what happens? And what happens is that, in fact, during the first week, if we give protein synthesis inhibitors after each recall, we disrupt the memory. So memory is reconsolidating to strengthening. 2 weeks later, there is no strengthening, there is no disruption. Memory is consolidated. By giving this 3 retrieval, we can also prevent forgetting. In fact, 3 retrievals tested 2 months later compared to controls. As you can see here, again, there is a significant memory strengthening. So it works also not just at the first week, but over time. Memory remains stronger. And it's because of this reconsolidation, these retrieval-dependent protein synthesis processes that strengthens the memory. But they have to be given when the memory is young. So if we look at the curve, retention curves of these animals, we see this is what happens over time. The blue one is the controls. They get trained, and then we just test their retention at different time points. So memory gets stronger over time, and then it gets weaker again. They start to forget. The red ones, they got the same training, but they were reminded 3 times during the first week. So there is a significant memory strengthening. We don't know if this could be here. We don't have this time point, but certainly remains stronger over time. So we prevent forgetting by reminding, through reconsolidation. So that's what reconsolidation does. Strengthens the memories. And it is temporarily limited. So then we thought, okay, let's strengthen old memories. When we forget them, can we strengthen them again by reminding these animals? So we give the same 3 reactivations, recalls, but now 4 weeks after the training. And I showed you that at that time memory is not labile anymore, but perhaps we can strengthen it. So we did that. Animals are trained, 4 weeks later they get 3 reminders, and this is the control group. Then when we test the animals after the 3 reminders, actually they had less memory. What happened? Oh my God, we wanted to strengthen the memory. But actually, 4 weeks later, we know that reconsolidation doesn't occur. So maybe this is extinction, and there is a way to test that. we're going to give them a reminder, food shock in a different place, and if that's extinction, memory comes back. And that's exactly what happens. These are the same rats. After the reminder, food shock, memory comes back. It's extinction. So then we repeat it again in the control group, the same thing. 3 reminders, memory drop, give the reminder shock, memory comes back. It's extinction. So memory is very dynamic. There is no fixation. There is no storage fixated. Changes with time. A young memory with reminders strengthens. A mature memory with reminders, if it is consolidated, it remains consolidated, and there is no change in the level of strength. An older memory, when we start to forget, softens. So the reminders actually leads to extinction. Beautiful. We have to keep that in mind for treatments, right? Because if we want to induce reconsolidation and disrupt reconsolidation, but we actually induce extinction, that's not the way to go. Maybe we want to induce extinction and strengthen extinction, not reconsolidation. Those are two different mechanisms, two different processes. So this reconsolidation, even for post-traumatic stress disorders, has still a long way to go. We need to understand temporal phases, we need to understand the strength of the memory, we need to understand the age of the memory, and there are beautiful data, but they are all done at very short time points. A day later, 2 days later. Yeah, that's the phase during which the memory is consolidated. But in clinical settings, that's not when the patients come and see you or a psychiatrist. Okay, a little bit about mechanisms. If we want to go into pharmacology and maybe have a combination of behavior and pharmacology to speed up the process, I'm going to show you that one way perhaps to look into is to look at the stress mechanisms. Well, you are very familiar also in your treatments with that. So, nothing new, obviously so. We know that emotionally arousing events are better remembered, they create long-term memories, so these mechanisms are important for the consolidation and the reconsolidation. They must be important for the reconsolidation of the memory as well. And that's what we tested. Stress hormones are known, noradrenaline, cortisol. We focused on cortisol, actually corticosterone, because we are in the animals. We know that stress leads to the activation of the hypothalamic pituitary adrenal axis, release of cortisol. This acts in many systems, prepare us to flee, and a number of defensive reactions, as well as it releases, it brings cortisol to the brain, and the binding to this cortisol receptor, glucocorticoid receptors in the brain, have an important effect, including memory strengthening and modulation. Number of studies have shown that a little bit of stress is very good for memory. We need that. Too much stress is bad for memory. This is importantly going through glucocorticoid receptors, the receptors for these hormones, steroid hormones. So we asked the question of whether if we block these receptors, the receptors for cortisol in the brain, can we disrupt these memories? Can we weaken them? And we can act on consolidation or reconsolidation. In this case, we're going to show you reconsolidation. So we're going to use an antagonist of this receptor. It's called RU486, known also as mifepristone. And we're going to give that after the retrieval of the memory. So it's the same thing. They are trained, 2 days later they are tested, they record the memory, and then they are injected with this mifepristone. It's actually a systemic effect in this case because we need to show whether this can work in clinical settings. So when they are injected with mifepristone, if they are tested again 2 days later, the memory here in the red bar, test 2, is significantly decreased compared to controls, and compare also to animals that received the same RU injection without the retrieval of the memory, which is the yellow bar. I lost it here. So it has to be the combination of retrieval and pharmacology to disrupt the memory. The memory remains disrupted if we test them again at later time points to see that the effect is persistent. It doesn't come back if we give them a reminder shock. It's not extinction, it's actually gone. But then we look at whether the same treatment can affect the memory of a higher shock intensity. So if the memory is more traumatic, can we disrupt those memories? And the answer is, we couldn't. Memories were strong here. We tried different concentration of the drug. However, this stronger traumatic memory can be disrupted if we use protein synthesis inhibitors. So this drug is not strong enough, and also it interferes with stress. So we thought that perhaps an intervention that is too early, 2 days after this very shocking experience, is not sufficiently blocking the receptors. So let's give it some time. We cannot give too much time because memory becomes consolidated. But let's give it a week so that the stress levels off a little bit. And we're going to give 2 treatments just to see if we can see some effect. So, and what we found, that this is the 2 days that I just showed you. There was no decay in memory after treatment. If we wait a week and then we give the retrieval followed by the pharmacological treatment, now the memory is weakened. And then again, that depends on the retrieval. We have to give pharmacology together with the behavioral. And it remains weakened, and it doesn't come back with a reminder food shot. So there is a possibility to intervene, but again, we have to take into account, and this drug can be used in clinical settings, it is approved, but we have to take into account timing, stress levels, when we do the intervention. It has to be not too soon, not too late. And then we tested how many treatments do we need to give in order to have the maximal effect. And we found that 1 treatment actually does the most effective disruption. The second treatment decreases a little bit, but then there is no other further decrease. So it's actually a pretty effective way to disrupt the memories. Only 1 to 2 treatments if we use the combination of pharmacology and behavior. But this type of treatments can also be taught with behavioral type of interferences. Pharmacology does not to be the only way. It's not the only way. So one possibility certainly that may interest psychiatry is to look at interfering with the stress pathway, particularly the glucocorticoid receptor-dependent pathway. We actually know now much more about the mechanisms that are induced by this glucocorticoid receptor activation in the brain. That's what we study in the lab. To weaken memories if given after retrieval, and also if given after training, so we can interfere with the initial consolidation as well. Let's switch it around. We have disrupted all these traumatic memories. Now, can we use the same knowledge to enhance memory? Since reconsolidation strengthens memory, can we find the mechanism that we can tap into and then strengthen the memories? And the answer is yes. So we have identified the mechanism of consolidation in the hippocampus. Turns out that one factor that we found required for long-term memory formation, consolidation, is insulin-like growth factor 2, a growth factor that is important during development, but was very little was known in the brain. We found that it's regulated after training, and we found that if we give IGF-2 in the hippocampus after training, we actually very significantly strengthen the memories. And the effect is sustained over time and also prevents forgetting. So if we test weeks after the treatment, we see that the memory is still strong. We also can strengthen these memories if we give IGF-2 after retrieval, not only after training. So if we wait some time after training, we give IGF-2, there is no effect. It has to be combined with an active trace. But if we retrieve the memory, say 24 hours after training, and we give IGF-2, then boom, the memory can be boosted again. So consolidation and reconsolidation are phases of strengthening. consolidation. It's an overall consolidation process that lingers for some time. So training induces these phases of consolidation, the initial one, reconsolidation with reminders, reactivation of the trace, and these reminders that leads to strengthening and stabilization and consolidation, they can be explicit, but they can also be implicit. Dreams, sleep. We don't know what happens during. I show you that time. The animals that stay in their own cage do nothing. They strengthen the memory over the first week. How does that happen? And then they forget. So we think that actually reconsolidation is a phase of this lingering consolidation process that lasts for weeks, in this case. Other types of memories may be longer. We have seen that those patients lost memories of years before. Perhaps until the memory remains hippocampal dependent, it can be strengthened or weakened to this process. And that's our model. That's our conclusion. That's how we go about in studying the mechanisms. But importantly in this room, I think also what we need to remember is that retrieval, recall of the memory, opens up not only the strengthening process, but opens up the opportunity to make new traces. So the old one now can make new associations, and those are new memories. We consolidate, and those are new traces, new strata of memories that come up and give us a number of options in our behavior, our choices. So for the discussion, maybe I just summarize here a couple of points. The dynamic of the memory, storage, traces, the contribution of consolidation, reconsolidation, new learning, the contribution of the age of the memory, the contribution of the emotional state, and obviously the intensity of the emotion. And I want to end by thanking the people who did the work that I presented. And these are very, I'm fortunate to have a group of people that are really fantastic in the lab. These are graduate students or MD PhD students or postdocs. Maria Milekic was a graduate student in the lab. She's now at Columbia. Steven Taubenfeld, postdoc in the lab. Sophie Tronel, she was a postdoc, and she's now back in France. She did the studies on the second order conditioning. Dylan Chen and Gabriella Polonini, who worked on the IGF-2 project. Justin Reisberg, a student in the lab, who worked on the glucocorticoid receptor part. And Carmen Inga and Elisabetta Muravieva, two postdocs in the lab, who looked at memory strengthening via reconsolidation, and obviously the founding agency. And I thank you for listening and being here.